Hi Kim 110. In this video, we're going to start looking at lipids. And so lipids are molecules that contain fatty acids or steroids, or a steroid nucleus. And you can see an image of a steroid here. These are typically organic solvents, soluble but not soluble in water. Lipids are kind of named after the Greek word for fat. So we'll be talking about fats, oils, waxes. Those are all types of lipids, in addition to things like steroids. And they are pretty important for cell membranes, vitamins, hormones, steroid hormones. And we're kind of going to go through them in some classes, kind of categorized as waxes, triacylglycerides, glycerophospholipids, sphingolipids, and then we'll get into some of the cholesterols as well. And I'm going to come back to this outline a couple of times. This is kind of how we're going to go through the items. We'll look at them in terms of either having a fatty acid component or being a steroid nucleus. And they are both considered lipids, but we'll see the majority of these items are going to be kind of made up of something that looks like a fatty acid. So something that looks like it's got this kind of carbon backbone with a carboxylic acid tail, a functional group. And so within that, we'll see waxes, triglycerides, glycerophospholipids, and even sphingolipids. And then separately, we'll have kind of our steroid hormones. And so we'll kind of have a brief overview of what these are and then dive into each of them kind of more in depth. And so many of these, especially the triglycerols and the glycerophospholipids, will have kind of this glycerol backbone. And this is going to be typically a 3-carbon backbone that will have kind of 3 hydroxyl groups hanging off of it. And then what we'll see is the hydroxyl groups will get rid of the hydrogen. And then we'll have an ester being formed with fatty acids. So we'll see this actually forming something that looks like this. And then a fatty acid tail. And then over here, we'll have something similar, except for here would be our 3-carbon backbone. Except for here, instead of kind of having a carboxylic acid joined on to make an ester, we would have a phosphate group. And then something like with an amine alcohol or something else. Maybe even something that looks similar to what you might recognize as an amino acid that might have otherwise been used in building a protein. And so we'll see what these look like kind of in more depth in future slides. We also have something like a sphingolipid backbone. So instead of that 3-carbon with hydroxyl groups, we have a sphingolipid. And so this is actually the outline of a sphingolipid. And kind of in this orange here, it's curved. Or I should say that's the sphingosine curved there. And the sphingolipid shows how this could be a fatty acid coming off here. And then maybe some sort of nitrogen or amino group or something, coming off in other directions. Sphingosine is typically something that's got like 18 carbons in it. And then we see more of an ether instead of an ester. And then, of course, we will see some steroids as well. When we get into those in a separate video, what we see as a steroid nucleus will have some six-sided carbon rings. Notice that these are not benzenes. There are no double bonds here. So it's not a benzene. It's just a normal six-sided carbon. And so typically we've got 3 of those joined and then a 5-carbon ring hanging off the side. And then there will be some substituents hanging off of this, sometimes alcohol groups or ketones or some things hanging off of this kind of baseline steroid nucleus. So that is kind of the overview of kind of this outline, this flow chart of what our lipids look like. So they're either going to have something that looks like some sort of fatty acid backbone, even if that fatty acid looks more like a sphingolipid, or it's going to have a steroid nucleus. Okay, so let's start with these fatty acids and talk more about them. A fatty acid really is a carbon chain of different lengths with a carboxylic acid on it. And so a long unbranched carbon chain, typically 12 to 18 carbons long, it can be either saturated or unsaturated. So if you recall, saturated means that it's all carbon-carbon single bonds. Every place that could have a hydrogen does. Versus unsaturated means that it contains one or more carbon-carbon double bond. And so we typically draw them with the line angle structure, although you can draw it in the condensed form like that. We might even see it more condensed where we draw a bunch of these CH2s together and just say that, you know, there's 10 of them there. And then it is a fatty acid, so it is a carboxylic acid at the end. And then the line angle structure we'll be looking at a number of times here. So these are the same carboxylic acids that we've been looking at in the week past, but just much, much longer chains. And remember, these longer chains, of course, they have more than 5 carbons. So these are mostly insoluble in water. Although we've got, you know, some hydrophilic portion here, and we'll talk about, you know, in a not this video, but in a couple of videos from now, the potential for being amphiphobic. So, you know, water loving on one end and water hating on another, and why that might be important for cell membranes. Okay, so some examples of a fatty acid that's saturated with single carbon-carbon bonds all the way throughout. Lauric acid and coconut oil, you can see here with 12 carbons, counting with 1 here and 12 out here. Myristic acid is a nutmeg with 14 carbons. Palmitic acid is in palm oil with 16 carbons. Stearic acid is an animal fat with 18 carbons. And you can see their melting points grow higher the longer their chain is. So 44 versus 69 degrees Celsius. So here are some mono unsaturated fatty acids. So that means 1 carbon-carbon double bond. So palmitic oleic acid. So here is that unsaturated spot. So this is present in butter. Oleic acid present in olives and grapeseed oil. Here is that single, or I should say that 1 double bond. And we also have the potential for polyunsaturated fatty acids, where we have multiple sites of double bonding. So here and here for linoleic acid, linolenic acid, has 3. Arachidonic acid has 3 in a couple different spots. So we've got soybean, corn, meat, eggs, fish. Okay, and so if you remember back when we talked about alkene functional groups, those double bonds, of course we have the functional group of the carboxylic acid here, but in any place that we have an unsaturated item, we also have an alkene functional group, and of course then we could have a cis or trans. And if you remember, cis means same side and trans means opposite. And when we've got a cis versus trans double bond within these really long carbon chains, it's going to affect the layout in the 3D dimensional shape of these acids, of these fatty acid chains. And so this really kind of dives in to the health outcomes of eating and consuming these. And so here we can see unsaturated fatty acids drawn in the cis and trans isomers. So oleic acid here on our left versus oleic acid on our right. So oleic acid on our left is a cis double bond. So it's a monosaturated fatty acid found in olives with 1 double bond at carbon number 9. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so carbon number 9, and it is cis. And you see it gives us this kink. Okay, so that kink is really important. So that same side means that the chain is going down on both sides of my double bond. So if we remember back to the alkene section, cis, same side, versus trans, one kind of goes up and the other kind of goes down. So in essence, this kind of stays straight across as I'm drawing this line angle. As I'm doing a zigzag up and down of this line angle structure, it really doesn't affect how the overall chain lays out on our right side here for this for this trans double bond. So the trans isomer of oleic acid has a different name. It's oleic acid, and its 3D structure is a more straight layout. It's a straighter layout. And this actually plays a role. So we're going to come back to this, and we're going to come back to this in terms of what it looks like for health and how it can stack and things like that. But keep in mind, cis has kind of kinks, and trans is kind of straight. And think of that in terms of what it might mean for stacking, especially when you might have more than one double bond. Okay, so humans, though, are capable of making and synthesizing some fatty acids from carbs and other fatty acids in our diet. But we cannot synthesize sufficient amounts of polyunsaturated fats. So the ones that have multiple double bonds in them, like linoleic acid, linolenic acid, or arachidonic acid. So in those cases, we have to consume them. We will call those essential fatty acids. So essential means that we have to get it in our diet. We can't make it. No matter how much other fatty acids we eat, no matter how much carbs we eat, we cannot make these items that have multiple sites of double bonds. And so if we need it in our diet, if we need it in our body to do anything, especially for things like brain development, we have to have it in our diet. So particularly for a fetus, growing infants, young children during brain development, they can't create these. The mother can't create it. It's got to come in from the diet. Okay, so now that we know what saturated versus unsaturated are, and now that we know what humans can and cannot create themselves, so must eat, let's talk a little bit more about saturated and unsaturated. So saturated fatty acids, again, contain only carbon-carbon single bonds, and they really fit closely together in a really regular pattern. So there's some strong dispersion or van der Waals forces between these carbon chains, and those are the ones that are kind of hard for us to visualize, those intermolecular forces that are really instantaneous, really weak, but also going to kind of keep this stacking pattern together. So if you imagine that these saturated fatty acids can lay on top of each other as shown like this, they're going to stack really easily. And when they stack and when they layer, imagine just that. They're stacking and they're layering together. And so you might see something that's stacking and layering like this. And even in some cases, a cis double bond may do the same thing. Let me back up. A trans double bond may do the same thing. So be better than what I just said. A trans double bond, where you still have kind of this straight, nice, flat layout, may do the same thing. But when you have now a cis double bond, where you've got kinks, especially if it happens to be a polyunsaturated item and they're cis, and you have multiple kinks, they'd really have to line up perfectly in order to stack. So if we look at oleic acid over here, you can see kind of this mess. They would really have to perfectly line up. Now, if I'm stacking these and I'm forcing them to line up, man, I could do it, but this is just biology and this is chemistry and this is just happening in our cells. So the likelihood of them, lining up perfectly in your bloodstream on their own isn't very likely, because they're not straight, because they have this kinked shape to them. It's not likely that they're going to organize perfectly enough to line up. They're probably going to be a mess like this, versus something that's unsaturated or has a trans potentially double bond. They, even if they're not perfectly, maybe one's over here to the right, one's over here to the left, they have a much easier chance of stacking. And so that means they also have a higher melting point than something of a similar mass. So fewer interactions can really happen in between something that can't stack nicely. And we'll talk about what that might mean in terms of cholesterols and what that might mean in terms of fat versus oil. Okay. So now we're going to move on to something called prostaglandins. And this is more of a hormone-like substance that really revolves around fatty acids in terms of how it's created. These are formed through a polyunsaturated fatty acid that's 20 carbon atoms long. And there is possible to have different substituents attached to different prostaglandins. But typically, our 20 carbon fatty acid will hook itself around and form a 5 carbon ring. somewhere in about the middle, and we'll see that in a moment. It can have many functional groups, so it can raise or lower blood pressure, it can stimulate contraction and relaxation of muscles of the uterus, for example. So here are some examples of pictures of just a couple of prostaglandins. So prostaglandin E and F. Okay, so in fact, let me actually skip forward to this image. So here is arachidonic acid. So here is kind of our starting point. And so this is kind of what we would get with pain. So this is one of our pain inducers and inflammation inducers. And so we start with arachidonic acid. And so if you notice, we would normally see this chain kind of continue off and to the left. And we just kind of, for purposes of showing this in a really easy way, are just turning this over to the right. And so that's okay. But we would, I think we've even seen this a few moments ago. Let me see if I can pull this up. Yep, so here we see it here. Arachidonic acid. So here we see it kind of laid out straight. And now we're just going to kind of curve it around here. Okay, so I'm just going to really quickly label our carbons so you know where we're at. So carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, oops. 13, 14. Okay. So what we're going to have is carbons number 8 and 12 coming around to form a 5-carbon ring that you see on the left-hand end of each of the kind of products here. And so this is how kind of you could be formed in a couple of different pain items, pain prostaglandins. And so we see that we could have, you know, a couple of different hydroxyl groups attached in some areas. And here we've got kind of an inversion of our carboxylic acid. Because, ooh, I'm sorry. Because of a double bond here that's not existent here. Here we've got a ketone, a hydroxyl. So we've got a number of different types of prostaglandin. Each of these 3 items are involved in inducing inflammation and sending pain signals, but all of them have been formed from arachidonic acid. Oftentimes, that is the kind of function of an NSAID, so a non-steroidal anti-inflammatory drug. That's an NSAID. These block production of prostaglandins, and so therefore that's how NSAIDs work to decrease pain and inflammation. And so if we take some sort of NSAID, such as Aleve, for example, we can essentially block the formation of a prostaglandin from arachidonic acid. So some examples of NSAIDs, Aleve, naproxen. We'll have some work on this in your lab activity, which I think I will actually have you do now. So before lab, go ahead and get into hands-on lab and complete exercise 1. It's the comparison of NSAIDs with, that is within the hydrolysis of acetylsalicylic acid. So go ahead and find that lesson and do that exercise 1. And you'll probably have to do the pre-lab work first. Okay, let's continue on with waxes. So waxes are typically naturally found on the surface of fruits and leaves, particularly so that they don't lose moisture to the atmosphere around them. And so the idea there is that, they uptake, the plant can uptake water at its roots. It wants to be able to keep that in. Waxes can also be found on the skin, fur, feathers of animals, and hopefully provide some waterproof coating as well. And so here are some images or some structural components of some beeswax, jojoba wax. So here we see esters as the center of these waxes. And then, 18 to even maybe 30 carbons on either side. And so the way these work, in the body, our fatty acids are stored as something called triglycerides. Triglycerides are an ester of glycerol and basically 3 fatty acids. And we kind of already looked at this a little bit at the beginning of the video. A triglyceride is going to be formed when kind of a 3-carbon hydroxy group is esterified with 3 different fatty acids. And so this is a pretty good image of what that looks like. And in fact, if we go forward one more slide, we can see that broken down even further. So here is what we call a glycerol. So it's 3 carbons, a chain of 3 carbons. Each of them has an alcohol group on it. So each of them has an OH on it. And then we join each of those OHs, each of those alcohols, with its own fatty acid chain. And so you notice that kind of all of the fatty acids that we'd had drawn so far, I think the carboxylic acid was on the right-hand end. So now flip it over. So this is the same thing we've seen so far, but the carboxylic acid is on the left. And that way we can look at it in terms of an esterification. So we saw esterifications in the carboxylic acid ester chapter. This is the same reaction, but it's going to happen each time. So it's going to happen at this alcohol with 1 fatty acid chain. It will happen at this alcohol with a different fatty acid chain. And it will happen at this alcohol with a 3rd fatty acid chain. And interestingly, depending on what's going on in the body or what's being built and stored, it can be 3 of the same type of fatty acids, or it can be 3 completely different fatty acids. All 3 of them could be saturated, all 3 of them could be unsaturated. It really doesn't matter. It really just depends on what the body needs and what the body has available. But what we're doing here is just an esterification bond. So we're taking an alcohol in 3 different parts of the same molecule, and we're taking 3 separate carboxylic acids and joining them together. And overall, we have what's called a triglyceride with 3 different ester bonds in it. And in doing so, we've pulled out 3 water molecules. So if we go back to the slide before, what we're looking at up here is just something much more simplified. So here was kind of that glycerol backbone, and then 3 different fatty acid chains hanging off. And again, we could have different fatty acid chains. So here's stearic acid. So we've got kind of, 16, 17, 18 carbons. Here we've got oleic acid where we've got a double bond here. Here we've got palmitic acid. And so we can have a mixed triglycerol, and that's okay. So triglycerols are really the major form of energy storage for animals, especially animals that hibernate. They eat a lot of seeds, plants, nuts that are really high in calories. As the outside temperature drops, the animal goes into hibernation and the body temperature drops to nearly freezing. And so cellular activity, respiration, and heart rate decrease. And so that really means that we can't necessarily go through the Krebs cycle as normal. So the energy sources as normal. And so during hibernation, really their stored fat in the form of triglycerols is really their only energy source. And interestingly, we'll see that it's also in some cases some of a little bit insulating as well. Okay, so that kind of makes me sometimes wonder what's the difference between a fat and an oil. And so a fat is something that's usually solid at room temperature. It comes from animal sources like meat, whole milk, butter, and cheese, versus an oil, which would typically be liquid at room temperature. So we usually get this from some sort of a plant source. So simply speaking, fat, solid at room temperature, oil, liquid at room temperature. But in chemistry, that's a little bit too simple. So oils like from olive and peanuts are typically mono unsaturated because they contain a lot of oleic acid. Corn, cottonseed, safflower seed, sunflower seed are polyunsaturated because they contain a lot amounts of fatty acids with 2 or more double bonds. Palm and coconut are solid at room temperature because they have a lot of saturated fatty acids. And so that tells me then that something must have to do with whether or not saturation, double bonds presence or not, must have to do with oil versus fat. And so saturated fats do typically have a higher melting point compared to unsaturated fatty acids because they pack together more tightly. And so these are going to be the ones that are usually found in animal fats, coconut oil and palm oil. And that has to do with that stacking. So if it's saturated, and if we can go back to the images from before, so if it's saturated, it's really going to be able to stack easily versus unsaturated, particularly in the case of cis bonds, it's really going to be not stacking nicely. And so we'll have typically a liquid at room temperature. So that brings us to something called a hydrogenation reaction. And this brings us again back to the alkene chapters. So those double bonds in an unsaturated fatty acid reacting with hydrogen gases to produce single bonds. So again, a carbon-carbon double bond, which typically would be also connected to more carbons on either side, and probably a hydrogen as well. And so that means that we're going to break part of this double bond and bring in more hydrogens. So this is hydrogenation. So a hydrogen gas is bubbled through heated oil, typically in the presence of some sort of nickel catalyst. And so a hydrogenation reaction is often used commercially. So an addition of hydrogen is stopped before all the double bonds of a liquid vegetable oil become completely saturated. And so we would call this partial hydrogenation of a liquid vegetable oil changes to a soft kind of semi-solid fat. So that's like our Crisco or margarine, for example. So a more saturated product with a higher melting point. And so we can have something like an unsaturated fatty acid can be cis with bulky groups on either side, trans with bulky groups on opposite sides. And so during hydrogenation, those double bonds are converted to these single bonds. But a small number of the cis double bonds can be converted to the more stable trans, causing a change in like this overall structure. So a small number of those kinked cis double bonds, so the ones that would have looked like that, might be converted into the more kind of flatter, kind of keeping it straighter. So this structure change. And so in the body, these trans fatty acids, the ones that are straighter, they look and behave a lot like saturated, the ones that don't have a double bond at all. And so they're estimated to be about 2 to 4% of our total calories. And so they raise our LDL cholesterol and actually lower our HDL cholesterol. So they're typically considered to be pretty bad for us. And so here's an example. So here's our cis fatty acid. And here is this kind of commercial hydrogenation idea. And so some of it can be kind of going all the way through to the saturated product. And some of it goes through kind of what looks like a trans fatty acid. And so that's gonna be what we're looking at here, 2 to 4% of our total calories in this trans fatty acid. And so foods containing naturally occurring trans fatty acids, though, like milk, eggs, and beef, and deep fried foods, bread, some baked goods, cookies, crackers, chips, vegetable shortening, margarine. These are the ones that we kind of often consider not so great for particularly our cholesterol, our health. We also have something called intraesterification, which is a newer process. And this can be used to change an unsaturated vegetable oil into products that have properties of like a solid and semi-solid fat, saturated fat. And so this is a little bit different than what we just talked about. So intraesterification is used to hydrolyze these ester bonds in the triglycerol. And so that's that 3-carbon backbone with all these esters and the fatty acid tails. So of these vegetable oils using something called a lipase enzyme, so that the glycerol and the fatty acids are formed. And so if we move forward, what we're looking at then is kind of during this intraesterification, an unsaturated oleic acid in a vegetable oil. So unsaturated, remember, has some sort of double bond in it, could be replaced by a saturated stearic acid. So we can kind of just replace the whole acid. And so here, for example, we can take a double bond and replace it with something that has a single bond. And that gives it a higher melting point. And we're talking about this being done in food sources. And so you might be looking at something that, a shelf life or packaging, something that's more packaging friendly. Okay, so let's talk about something more along the lines of what happens when we eat these. So we do need to eat fats, whether it's, produced or, eaten naturally. We do also need to store fats and then use them later. So when we've got a triglycerol stored in our body or in our diet, we need to be able to get it apart. And so if you recall, if I jump forward here, here's our triglycerol, and here is this glycerol backbone. It had an OH group on it. And then, of course, this was a carboxylic acid, fatty acid. And so now we've got this ester bond. And what we're going to do is we're going to hydrolyze it, put water back into each of these bonds, and separate each alcohol component back apart from each carboxylic acid component. And so if you recall from the previous chapters, we can do that either through an acid hydrolysis or a base hydrolysis. And if we use a base, then we call it saponification, where we do that with a base, and the reaction of a fat and a strong base usually makes some sort of carboxylate ion salt, and that is how we form soap, if you recall. And so we'll be making soaps in lab. So saponification and soap. Here we've got our fat or oil of some sort. So here's each ester. And we're going to break it apart. We'll be left with a glycerol, our triglycerol group there, and then our carboxylate, so what used to be our fatty acid tail, now is an ion, and then sodium from our NaOH base. And then we'll have 3 of those, of course. Okay, so that's where we're going to end this video, and we'll come back to look at the rest of, or I shouldn't say the rest, but some more of the types of lipids from our flow chart here in the next video.